And our next speaker is Dr. Daniel Di Toro from the Brigham and Women's Hospital, Boston, Massachusetts General Hospital in Boston, and Harvard Medical School and Ragon Institute of MGH, MIT, and Harvard in Cambridge, US. And the presentation is on acquired resistance to FGFR inhibitors.
Thank you. Thanks everyone for coming. Thank you to the AMMF for the invitation. It's a pleasure to be here. My knowledge of cholangiocarcinoma is very, very, very narrow, and it's really nice to be here and be exposed to so many different perspectives and ideas, learning a lot. Also, I did my GCSEs in Aylesbury in the 90s, and I haven't been back since I was 14, so it's really been a great trip. I'm going there tomorrow. For those of you who don't know me, and that's everyone, my name is Daniel. I'm a molecular genetic pathologist at Brigham and Women's and Mass Gen. I'm an immunologist by training. So if anybody wants to talk about T cells later, just say CD4 and I'll start going. I don't want to just bore you with a list of details from all the papers out there today. So I'm going to give a relatively superficial description of what is happening so that I can focus on why I think that's happening. So cholangiocarcinoma, as we all know, it's a disease of bile duct epithelium, cholangiocytes. It is a rapidly fatal disease, and it responds poorly to traditional chemotherapy. It's also remarkably heterogeneous. There are different diagnostic subcategories based on anatomic location. A lot of this has to do with developmental origin. And each of these groups is defined by multiple subgroups, multiple molecularly defined subgroups. Now, this discovery has led to a transformation in patient care. In the US, patients with intrahepatic cholangiocarcinoma, about 50% of them are getting targeted therapies now. I specifically study intracellular cholangiocarcinoma, and I study FGFR altered cholangiocarcinoma. Why focus on such a narrow slice of the spectrum? Well, it's a very difficult nut to crack, and I think that if we can solve this problem, the lessons from that problem will be generalizable to other problems. I think by fixing this, we can fix other things. And so this is where I put my efforts. So what is an FGFR? It is a fibroblast growth factor receptor. There are 4 different receptors that are very, 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 very similar. It's very difficult to make a drug that targets only one of them. In cholangiocarcinoma, it's almost always FGFR2 that's altered. Usually what happens is the FGFR2 gene gets rearranged and stuck next to a different gene. But you also see amplifications, and there are certain activating mutations. Regardless of the alteration, all of them lead to increased signaling downstream of the receptor, and it is this dysregulated signaling that drives the cancer. So if we know that the dysregulated signaling is driving the cancer, then if we stop the dysregulated signaling, we should stop the cancer. This idea has worked reasonably well in other tumor types. So as a result of this, a large number of drugs targeting the FGFRs have been developed in the US. INFI, PEMI, and FUDI have been approved for use in iCCA patients with FGFR2 alterations. There's a whole bunch of other drugs that are in clinical trials, and there's yet more that are being developed in preclinical studies. In fact, this paper was published 13 months ago, and it's already out of date. So this flurry of activity, it's very exciting. It bodes well for patients. It's wonderful. But they don't work that well. Response rates are around 20 to 40%. Even the best one is about 40%. And among patients who do respond, the duration of response is only 6 to 10 months. Compare this to other diseases where kinase inhibitors can achieve 50 to 95% response rates and durations of response that are measured in years. We have a long way to go. Now, we know in the lab that the drugs work. We can look at purified protein and we put the drug in there and it doesn't get activated. So if the drugs work, then why don't they work? Well, resistance to targeted therapy, to any therapy, can be categorized conceptually into 2 different groups. patients who do not respond to the drug are said to have intrinsic resistance. They have a large number of cells that have some set of properties that confer resistance to the treatment before they're even exposed to this. Because those cells are already resistant, the tumor doesn't really shrink, it doesn't respond. Those are our intrinsic resistant non-responders. In contrast, patients who do respond but then progress are said to have acquired resistance. In these patients, it's thought that the cells are initially, tumor cells are initially sensitive to the treatment, but then some of them pick up a property that confers resistance, and then they grow out and the tumor grows again. Now, in reality, it's not quite this simple, it's not this clean cut. For example, it's possible and even likely that the cells with acquired resistance actually do exist before exposure to the treatment. They're just at such small levels that it takes a very long time for them to grow out. But the groups are different in important ways, so it is a useful categorization. For example, patients with intrinsic resistance do not have kinase domain mutations in the target. Most patients with acquired resistance do. So there is a categorical difference between them. We're focusing on acquired resistance. If anybody knows what's driving intrinsic resistance in these patients, please tell us. We can't figure it out. It's certainly not to anything obvious. So I want to try to explain to you why we're seeing the patterns we're seeing. In order to do that, I'm first going to have to teach you some basic biochemistry. We're going to talk about kinases. So every single kinase in your body, every single one has an N lobe and it has a C lobe. They're called this because proteins are synthesized as strings, and one side has an N terminus and the other side has a C terminus, and they go plop, 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 and they fold together and they become this like globule, and they got a C lobe and an N lobe, close to one end, close to the other. Okay? For an enzyme for a kinase to become activated, the N lobe has to go close to the C lobe, and it does that by pivoting around the hinge region, okay? This bulky alpha C helix comes down towards the N lobe, and this alpha activation loop pivots up, comes in proximity to the C helix. These movements create a pocket that allows substrate and ATP to get near the enzymatic portion of the kinase, and ploop, you take a phosphate from the ATP and stick it on the substrate. You activate your signaling. Now, we want the kinases to be very, very, very sensitive to changes in ligand concentration. So we want to make it really easy for them to be activated. But inappropriate activation causes cancer. So we also want to make it really hard to activate them. How do you make something really easy and really hard? Well, I think that the answer is tension. You create tension. If you imagine a rubber band hooked to a post by a pin, and you pull on it, and no matter how hard you pull on it, it doesn't go anywhere. And if you flick the pin up, boom, it snaps and you get movement. So we want to create tension. And the way we do this with kinases is this thing called a molecular brake. There are 3 residues. One is right here in the hinge, one is over here, one is down here. And they form hydrogen bonds with this little bit in between the beta 4 strand you can see here and the C helix right here. It forms a hydrogen bond, and it holds that N loop up so that it can't go down. There's this tension. It's pulling it back, okay? Can't get the C helix and the activation loop, don't get activation. In FGFR2, the variants or the residues that are doing this are N550, E566, and K642. And importantly, from multiple studies, we know that this is the one that's really doing all the heavy lifting. So just remember those numbers. You can also make it so that only certain things can pop the pin, right? We can do that on the ligand end, but we do it intracellularly as well. There's always a residue called a gatekeeper residue, and that's going to be, it's the first residue of the hinge, and it sticks out and it blocks access to the back pocket. So only certain substrates can fit in there, okay? You get specificity. Only one finger can pop the pin. And in FGFR2, it's V565. This is from a relatively recent relatively recent paper. This is a composite of 3 different studies. These are mutations in the kinase domain that we find in patients who have acquired resistance to FGFR inhibitors, okay? First of all, what stands out is there's a lot of them. Okay, there's a lot of them, and they're happening at they seem to be distributed almost randomly throughout the kinase domain. This blue part is everything we just talked about before. That's our kinase. So we got a lot of mutations. They're distributed almost randomly. There's got to be some reason of why they're showing up. Well, let's take a look. N550, E566, K642. Note, the numbers are all off by 1. It just depends on what reference transcript you use. So that's our molecular break. That is our gatekeeper. K660, I didn't show you, but it's in the middle of the activation loop, and it's really important in maintaining kinase activity. The next two most frequent mutations, at least in this data, M538, L618. Again, I won't bother with the specific mechanisms. They're a little more complicated, but these ones also stabilize the inactive conformation. They're part of that rubber band, pulling it away from being active. So every single one of these mutations directly relates to the function of the enzyme, of the kinase. Now, there is some evidence that these gatekeeper mutations also have steric effects, that the molecular brake mutations have steric effects like a gatekeeper. And at least in FGFR3, there's evidence that the gatekeeper mutation mutations that also activate the enzyme. So it seems like they're both doing both. And I want to suggest that perhaps it's better to think about these mutations as existing along two spectra, one describing their steric effects and another describing their effects on signaling activity, and that perhaps the clinical frequency of a mutation is a function of the net effect along these two functional spectra. I hope that makes sense, because I think that's what's going on. But the problem is, that data, that's 3 very small studies. There's not a lot of data points. We don't actually know the true distribution. I think it makes sense. I think it will bear up, but we don't know that. We need larger data sets. This is a larger data set. This was recently published by Lipika Goyal, who was involved in 2 of the other studies. This is thousands of patients with bile duct cancers. Importantly, we don't know if it's cholangiocarcinoma. We don't know if it's acquired resistance, but since no patients have been shown to have kinase domain mutations before treatment, these mutations probably reflect acquired resistance, right? And we see basically the same pattern. We see N550, V565, E566, 618, K660, all the same ones are there. So we're probably getting close to understanding what the true distribution is. But while some of these mutations have not been studied in labs, we don't know their activity, many of them have, and we know that every drug has a different profile of activity against different mutations. As such, we would expect that every drug has a different resistance profile. We don't know what drugs these people took, so even though there's thousands of them, we can't tell you what the drug-specific resistance profiles look. So we need larger data sets that are better annotated and that are specific to patients with acquired resistance. And it's not just that, there's more complexity, there's more issues. Cholangiocarcinoma patients appear to develop polyclonal acquired resistance. This is one patient who, after treatment, here's pre-treatment, they have this ARID2 mutation, and they picked up 5 different mutations, including 2 different ones affecting N550. Here's another patient, 5 acquired resistance kinase domain mutations, V565, 642, 550, it's all the same ones. But they had 5 of them at the same time, 2 people with 5 of them at the same time. It doesn't happen all the time. Same study, 1 patient, 1 mutation, okay? And it's not just us, this sort of begs the question, are all of these mutations occurring in the same cell? Is this like compound resistance? If you're gonna make a drug, does the drug have to be able to target all these mutations at the same time? Or are these patients developing simultaneously multiple distinct resistant clones that are growing? I don't know which sounds more bizarre. So we did a rapid autopsy on one of these patients, and we did whole exome sequencing on a whole bunch of different lesions, and we looked for these mutations. And what we see is that some lesions don't have a mutation at all. Some of them have 1 or the other, and then one of them has 2. Now, it's really difficult to interpret this because we have so little data, but what I think is going on is that patients are simultaneously developing multiple distinct resistant clones. I think that this is probably a sampling biopsy. The biopsy probably, or a sampling error, the biopsy probably just picked up 2 resistant clones that had grown into each other. This is known to happen. I think that's sort of the simplest explanation. But then why are there some with no mutations? Maybe they had them and they lost them after we stopped treating them. But also, I don't know, maybe a third of patients with acquired resistance have no kinase domain mutations. So it would sort of follow that not all lesions need to have kinase domain mutations. And it's not just us that sees this. This is another group's work. We've got one patient here. Look at that. That's just, it's incredible. I mean, granted, all right, these are 2 different patients, sorry, but look at, we have N550, 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 4 different mutations affecting that residue. That is a really important residue. V565, V565, E566. That's a lot of mutations. This different patient, patient 7. Look at all these mutations. There's 11 that were detected. Now, a lot of them were detected at like very, very, very low levels. We don't know if they're really that biologically meaningful, but 3 of them were detected at very significant levels. This is amazing. The only other context I am aware of polyclonal resistance is when a patient is sequentially treated with different drugs. They pick up one, they get a different drug, they pick up another. I have never seen this in any other context. Why is this happening? What's different about cholangio or different about FGFR inhibitors? Something's different. As I mentioned, a lot of people have tried to study the activity of these mutations. There's a number of papers, a number of tables. I picked this one because it's kind of the most impressive. Don't worry about the details, the little small numbers. Pay attention to the colors. Those little small numbers are IC50 values, and IC50 value is supposed to tell us the activity of the drug against the mutation. Now, there's a lot of caveats to that, and we could argue about that for hours, but that's what they're supposed to do. Ostensibly, that's what they do. And we color these numbers according to what we think they do. Ooh, green is good, red is bad. There's a lot of green here. If there's so much green, why don't they work? Go take a look at the unbound free drug plasma concentrations in their phase 1 trial information. And then go take a look at the numbers here and ask yourself if we're drawing the right colors in the boxes. A lot of the plasma concentrations are lower than values we see in green boxes here, okay? And I think if you compare the plasma concentrations of unbound FGFR inhibitors to other kinase inhibitors, they're like an order of magnitude lower. These have very, very low achievable plasma concentrations. They did dose escalation. They tried to get higher doses. The patients couldn't tolerate it. So what I think is happening is that the very low plasma concentrations create a permissive environment that potentiates the emergence of a large number of resistance variants, some of which confer only a very modest effect. But that effect is meaningful when you can't go above a concentration. Just a 2-fold increase in IC50 could be the difference between responding and not responding. And you compare this to other conditions, like let's look at EGFR-driven non-small cell lung cancer, and you have T790M. That's basically it. That's the mutation that matters, and you have a concentration of drug that's like 10-fold higher, 15-fold higher. So it makes sense. If you can get really high drug concentrations, only the mutation that's really, 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 really potent will emerge, and so everyone has the same mutation. But if you have low drug concentrations, then lots of mutations can cause resistance, and so lots of mutations show up. So what do we do? Well, first, as I said earlier, we need to systematically examine intrinsic resistance. It's really important. We need to get response rates up. Second, we need to properly define the landscape of acquired resistance. As I mentioned, we're just sort of cobbling together small studies, and yeah, the distribution makes sense, but we need to know what that distribution actually is in order to make informed decisions, in order to develop next generation drugs. And we need to systematically examine cell-free DNA versus biopsy, and then hopefully with rapid autopsy and large numbers of lesions. So that we need to understand the clonal evolution. Am I right? Are we in fact independently acquiring resistant clones, or are they sort of forming together and then splitting? I don't know. We need to know. It informs drug development. And we need to actually figure out these mechanisms. I said what I think is happening, but we need to prove that. We need to actually know that. If we can do that, then maybe we can use all this information, we can integrate this information into a comprehensive model that explains the clinical landscape. We can use that comprehensive model to identify desirable traits in next generation drugs. And if we're able to develop that model, then maybe what we can do is we can use the same sort of steps to develop similar models in other areas and anticipate all of this. There's a lot of patients, a lot of people dying because we weren't able to anticipate which resistance mutations mattered. We should be able to anticipate which resistance mutations matter and make drugs that are able to deal with them from the outset. I think that should be the goal. It would save companies a ton of money, it would save lives, it would save me a lot of work. It's the right way to do things. And I want to close by addressing a theme that's sort of been present throughout the conference, which I'm really happy about. But this is a really, really difficult disease. It's very rare. None of us can solve it alone. I think we all know, at least we all know 3 patients, right? But if we all talk to each other, then we all know 100 patients. And if we all know 100 patients, maybe we can learn what's actually happening here. Unfortunately, the currency of science is authorship, and the only authors that matter are first and last. So we are incentivized to erect silos, barriers of access to our data. But I think that the currency of science should be citations. Citations are a direct measure of your impact on the field. Okay, that's what matters. And you get the same citations whether you're first or sandwich meat or last, right? I understand that accepting that and moving is risky, right? It's risky to voluntarily give somebody something of value that has value to you, but I think it's worth it. I think that if you focus on driving the field forward, that your career will be pulled along with it. I don't know that. I'm still early in my career. I hope that's what's going to happen because that's what I'm going to try to do. So please, everyone, talk, share your data, share your information, tell everyone what you think is going on, understand that it is risky, but do it anyways. I think that we'll do a lot better if we all do that. Thank you.